So, third point is homologous pair of chromosomes segregate during gamete formation. Fourth point is independently they move. Independent pair will move independently. That means, look here. Here there are two pairs. I'll write it as A and B. While moving, suppose A comes here, in another gamete B will go to another gamete. A chromatid will not follow B chromatid. A will go to one gamete, B will go to another gamete. That means independent pair will move independently. Here A moves to one gamete, B moves to one gamete. Here also C will move to one gamete, D will move to another gamete. So, like that, they told it is very similar to that. We can overlap this with Mendel's theory. But what is the speciality here? It is they use the word chromosome. Mendel was unable to use that word chromosome. But here, chromosomal theory, what it says, see, chromosomes are the vehicles of heredity. What Mendel told, all the characters are stored in the factors. Two identical chromosomes will be homologous pair. Two identical chromosomes will be the homologous pair. Then homologous pair will segregate. Factors belonging to the same character will segregate. That was the explanation given by the Mendel. Then independent pair will go independently. That the factors belonging to the characters will move independently of each other while moving. So, similar what Mendel told, same thing will be expressed by this. Okay. Now, next, I want to tell about the work of Thomas Hunt Morgan. You know, Morgan was known as a father of sex-linked inheritance. Thomas Hunt Morgan, known as father of sex-linked inheritance. He conducted an experiment and proved chromosomal theory of inheritance using fruit flies, Drosophila. Proved using fruit fly. Or Drosophila. Earlier it was known as Cinderella of genetics. This Drosophila was known as earlier Cinderella of genetics. Why he took Drosophila? Because they can grow in simple synthetic medium. They can grow in simple synthetic medium. They have short lifespan. The second important advantage of them was they have short lifespan. And breeding can be done throughout the year. Breeding was possible throughout the year. Hundreds of progenies per mating. They were able to produce hundreds of progenies per mating. Male and female flies can be recognized easily. What is the method to identify male and female Drosophila? See, this is the structure of diagram of Drosophila. This is female. This is male. You may ask, how can you tell this is male? Because you can see here a comb-like leg with a comb-like structure. I'll enlarge this. A comb-like structure will be there in its leg. It is enlarging. A comb-like structure. That is not there in female. It is easy to identify with the help of an ordinary microscope also. So that was the reason why he used. And another reason is males were smaller than female. Males were smaller than female. Males were smaller than female. So by naked eye we were able to identify, and because of that he used that particular organism for us experiment. Okay, then let us see about the new concept. I hope it is clear to you. Now what we studied, chromosomal theory of inheritance. Mendel's work was rediscovered by Carl Correns, Eric von Teschemark and Hugo de Vries. There are three scientists rediscovered the Mendel's work. In 1902, chromosomal theory of inheritance was proposed by Walter Sutton and Theodore Boveri. It said that pairing and separation of chromosomes are responsible for the movement of factors. Indirectly, they were telling that factors were they are situating on chromosomes. So, for the movement of factors, movement of chromosome is must. Then Sutton matched the movement of chromosomes along with Mendel's law of segregation. So he compared his idea of chromosomal movement with the Mendel's law, and he came to following conclusion. And now we study it as postulates of Mendel. What it says? It says that chromosomes are the vehicles of heredity. And two identical chromosomes will form homologous pair. What are homologous? Exactly mirror image. Even the internal arrangement of genes inside the locus also should be similar. Then Homologous pair will segregate during the time of gamete formation. Independent pair will move independently. So, this chromosomal theory of inheritance, in order to get its success, it needs proof. It needs proof. So, Thomas Hunt Morgan, he proved the theory of chromosomal theory of inheritance by taking Drosophila or the fruit flies as example. By taking Drosophila or fruit flies as example. So, they can grow in, why he took Drosophila? There was a reason. It can be grown in simple synthetic medium. Sweet solution or what we say, semi-solid paste of wheat, boiled wheat powder, along with some amount of sugar or jaggery is enough for the feeding. Short lifespan, they can live for a very short duration. That's about, they are able to complete their lifespan within 12 to 14 days. 12 to 14 days. That was another reason for their success. Then breeding was possible throughout the year. Then hundreds of progenies per mating was possible to produce more number of progenies. Male and female flies can be recognized easily. Males were smaller than female. And how to recognize? Thoroughly by observing by keeping them under microscope and observing their leg. You may wonder how it's a fly, it's having wings, how it can be seen under microscope. So we have to make them unconscious by treating them with ethyl alcohol. So we have to bring alcohol to the nest or to the bottle where we culture this Drosophila. In the bottle, that open mouth of the bottle will be covered with the lid of cotton which is soaked in ethyl alcohol. Then all the flies will become unconscious, and during that time it is easy. They won't die. They will become unconscious. After a few minutes later, after hangover, they will come back to normal life. Now, we'll study about linkage and crossing over. Another important concept of genetics. Linkage and crossing over. Dear students, there are certain genes which remain together for many generations. And such genes are called linked genes, and the mechanism is called linkage. It is physical association of two or more genes on a chromosome. Physical association of two or more genes together for many generations. Two or more genes together on a chromosome. A chromosome. They must be situated on a chromosome. On a chromosome. Okay? Physical association of two or more genes together on a chromosome and it will remain, and they will remain together. They will remain together for many generations. Many generations. Not one generation. Such genes are called linked genes, and the phenomena is called linkage, and the phenomenon is called linkage. Imagine that there are certain genes on a chromosome. Suppose this is a chromosome. Okay. Here there is one gene, A, and immediately very close to it there is another gene. I will mark it as B. Immediately very close to it there is another gene. I will mark it as C. In the first generation also their position was like this. After 100 generations, in this particular chromosome, they are located in the same place without any change. They are located in the same place without any change. After 100 generations, they are located in the same place without any change. And this phenomenon is called linkage. This phenomenon is called linkage. The disturbing factor of the genes to show this phenomena, all other genes can't show this phenomena. For example, there is another gene which is located here, that is called X. Another gene is located here. I can't say they are linked genes because they will not remain together on the same chromosome, or I will tell you, X is here and Y is somewhere here. In the next, after 100 generation, X will be here, Y, this Y may not be here. It may be another small y, or the position might have been changed. They will not attached with each other. So, small y may come here. It is not due to the mutation of this. The other from maternal, this is father's chromosome. Mother's chromosome will give small y to this, and that father's small y will go to mother. And this exchange of small y from mother to father chromosome and father's capital Y chromosome to mother, it is possible because of crossing over. What is that? Crossing over. You may ask, sir, immediately while seeing this video, suddenly a question arises in your mind. Sir, is it not possible to have crossing over here? Can't we exchange the father chromosome gene with mother's gene? It is not possible here because imagine that if you want to, imagine there is a thread and you are going to put 2 knots in that particular thread. If the knots are very close to each other in the thread, you can't form, or you can't put another knot in between. Otherwise, I will explain it like this now. 1 minute. I'll use this. Imagine that this is, I hope you are seeing this. This is one chromosome, and here is one knot. Okay. Yeah, here is one knot. Here is one knot. Can you see this? This is one knot. And this is another. They're almost very close to each other. Very, very close to each other. Is it possible? Is there any gap between these two? This is one gene. This is one gene. And this 0 is one gene. And this is another gene. How many genes are here? 2 genes. Okay. Is there any gap between them? No. When there is no gap, there is no question of separation. But if they are far apart from each other, then the chances of crossing over occurs inside in this position. So one crossing over, second crossing over. So chances of separation of this gene and this gene is 2%. This is one crossing over, this is another crossing over. But when they are overlapped with each other, when they are very close to each other, imagine, is there any chance of crossing over? Cutting. Is it possible to cut the gap between the 2 alleles or 2 genes? There is no gap at all. There is no gap at all. They're all almost overlapped. So, imagine that this is one gene, this is another gene. These, I will call this gene as linked gene. They'll remain together for many generations. They'll remain together for many generations. They won't separate. So this is called linkage. And there are certain genes which are far little apart from each other. Such genes are called, in between those genes, there is a space to separate them, and that process of separation during zygote formation, during exchange of paternal and maternal chromosomes, it is possible, and that exchange is called crossing over. That's why we are different from our parents. That's why we are different from our parents. Crossing over occurred. But when there is a birth of a newborn baby, people will come and see and compare the nose. It is very similar to parent, father, and ears. It looks like your mother ear. Then, in some it shows that some genes are not showed crossing over. They transferred as it is. I hope the concept is clear to you.